Hello. This this would be the luncheon symposium sponsored by the PerkinElmer. The title of the talk would be Non-Invasive PGT-A using PerkinElmer PG-Seq Rapid Non-Invasive PGT-A Kit. The speaker would be Dr. Gary Harton, Market Development Leader, Genetics, PerkinElmer. This would be the virtual presentation.
Welcome to my talk entitled Non-Invasive PGT-A using the PerkinElmer PG-Seq Rapid Non-Invasive PGT-A Kit. So I think the first thing to think about is understanding why we might want to use non-invasive PGT-A. So let's think pros and cons. What are the pros of non-invasive PGT-A? Well, it's technically less demanding than embryo biopsy. Certainly, I don't want to minimize anything to do with embryology and IVF, but certainly taking some media after an embryo has been moved out of a droplet is slightly less demanding than doing really good embryo biopsy. It's certainly more cost effective. The tools are cheaper. The time is less. It may allow for testing of more embryos. An embryo doesn't have to grow all the way to be a blastocyst for a patient to get genetic information about the full cohort of embryos to truly understand what's going on in their IVF cycle. And then lastly, it may satisfy governmental restrictions on embryo biopsy. There are countries today where embryo biopsy still needs to be approved. Each biopsy needs to be approved by a government body. And so obviously non-invasive PGT-A might get around some of those restrictions. Of course, if there are pros, there must be cons. The cons of non-invasive, there's contamination, which we're going to discuss quite a bit today. And then the concordance rate or accuracy of the test. And today, the gold standard of looking at the accuracy of PGT is invasive PGT-A with biopsy. So the concordance rate of a non-invasive to the actual biopsy is important to understand. So there are obviously some variables that might impact non-invasive PGT-A. Things like culture conditions, group versus individual droplets. How big is the droplet? What brand of media are you using? All of these things can impact the ability to find DNA and reliably diagnose embryos based on DNA that's found in the media. In addition, there's the risks of contamination. There's risk of maternal contamination and paternal contamination. There's a risk from an operator, be that someone at the IVF center, someone in between, or someone at the PGT lab end. The media itself can be contaminated, and we'll talk about that in a minute. We'll talk about a lot about contamination in this talk. From the lab, as I mentioned, from the embryology lab, the PGT lab, and then other sources along the way. There are other things in media, and again, we'll discuss some of those things through the talk today. So what impact does culture media have? So Polini in 2013 showed no PCR inhibition with blastocele fluid. However, culture media does contain a range of known PCR contaminants, including salts, EDTA, proteins, enzymes. All these things are known to get in the way of polymerase chain reaction. Therefore, any culture media that we use for non-invasive PGT-A, one has to understand what is the makeup of salts and EDTA and protein and enzymes so that you can design a whole genome amplification and a library prep and an analysis pipeline that understands those things may get in the way during PCR. And so it's a really important thing to understand the differences between taking a kit off the shelf that's meant for invasive biopsy and trying to make that into a non-invasive kit, or purchasing a kit from a manufacturer or developing your own workflow that's specifically around looking at spent culture media, because it is very different than doing biopsy. And it has to be taken into consideration that a bespoke kit made specifically for non-invasive PGT-A will likely generate better results than trying to shoehorn an invasive kit in and trying to make that non-invasive. So a variety of culture conditions have been published. I'm not going to read this slide to you. You can certainly have a look at these. You can see that some of these are spent culture media. Some of these are blastocele fluid with spent culture. Collection window is everywhere from day 1 to day 7. Various media brands, various droplet sizes, all fertilized with ICSI. Varying times that the embryo spent time in the media, and obviously varied results. And I think that's important to understand, is that all the little things, that decisions one makes along the way of developing a non-invasive program in your laboratory, each of those decisions can impact your concordance and therefore the results of the test. So as I mentioned, there are things in culture media that do get in the way of creating or amplifying DNA. So components of culture media can influence the size of the DNA fragments that are amplified during whole genome. And so here what we have is genomic DNA in culture media versus genomic DNA just in water, and then a no template control. And you can see that the size of the DNA fragments is different with and without culture media, and the intensity of the brightness of that smear is also different, telling you there's different amounts of DNA. So there's different sizes and different amounts simply by putting genomic DNA into culture media, which tells you the culture media can and does get in the way of, or can impact certainly, your whole genome amplification. Increasing the volume of culture media does negatively impact the WGA yield and fragment size. Again, this has to do with, this is looking at the number of microliters of culture media added to the WGA using genomic DNA in culture media. And again, what you'll see as more DNA or as bigger droplets are used, you get a more diluted basis of the DNA that's in the whole genome. So clearly, droplet size and the amount of DNA, or the amount of the amount of the culture media you put into the system, can impact your yields and therefore your downstream results. So this looks at then droplet size, so the amount of media the embryo was grown in, and during our development of our non-invasive PGT-A kit, we looked at droplet sizes anywhere from 60 microliters down through 10 microliters. For these, the same amount of the spent culture media was entered into the WGA, and you can see that you get highly variable results, not only from droplet size, but also from embryo to embryo and spent culture media to spent culture media, which then again, of course, tells you that non-invasive PGT-A in spent culture media can be difficult, specifically because you're amplifying DNA that's sitting in culture media that isn't ideal for PCR type tests. So variables that might impact non-invasive, I think the first one that we have to talk about is cumulus cells. Cumulus cells obviously contain maternal DNA. When cumulus cells are present, the medium can then contain a mixture of embryo DNA and maternal DNA, 46XX, which is going to lead, obviously, to maternal cell contamination issues. And it's something that we're going to talk a lot about over the next couple of slides. Just looking at media, not with anything grown in it, Hammond and colleagues in 2017 published a paper looking at a number of different brands of media and other things that go into culturing embryos during IVF, and looked for human mitochondrial DNA copies and human nuclear DNA copies. And you can see that there are varying amounts of copies, but the reality is that almost every single one of these solutions that is typically found in an IVF lab does already contain some human DNA. And so again, this has to be understood, that DNA from any other source other than the embryo is going to or can impact your results of your non-invasive test. And so it has to be understood that when you develop a kit for non-invasive PGT-A, developing it specifically for non-invasive, and to look for things like contamination and of the media from maternal-paternal, from the DNA source, from making the WGA work better, because even though it's spent culture media that has salt and protein and other things in it. Again, my thought here is that using a kit that was specifically developed in a large collaboration, specifically for non-invasive PGT-A, should long-term give you better, more reliable results. At least that's our opinion. So how important then is maternal contamination? This is similar to the chart I just showed a second ago, with some of the things sort of turned down, just to specifically look at the concordance of autosomal chromosomes to biopsy and sex concordance to biopsy, because in the end, most of the misses, the non-concordance here, a good part of them are going to be maternal cell contamination from the cumulus cells. And you can see in a number of studies, the concordance rates are all over the place, right? You'd like to see concordance rates, hopefully 100%, like in that one paper there, but that's not often going to happen. You're going to have some lower percentage of concordance, and contamination is a big part of that non-concordance for sure. So again, this is work from our development collaboration we did. It was 15 laboratories from around the world. It was a few thousand spent culture media samples we had in our hands. And what this is looking at is just sex discordance. So we know the embryo was XY, but the media came back XX. And you can see in some laboratories it's quite low. It's 3% in lab 1. It was 6% in lab 8. But you can also see that in lab 5, it was 53%. In lab 6, it was 47%. And so obviously a test where you're getting sex discordance and therefore non-concordance is going to lead to samples where you get the answer wrong. And that's obviously something that needs to be minimized as much as possible. Again, this is from our development study, all unpublished, but I think this really nicely shows that laboratory to laboratory variance, most likely in embryo culture conditions and how embryos are cultured and how the media is handled and sampled and processed, all of those things lead, we think, to increasing levels of maternal cell contamination and therefore lower concordance rates between biopsy and spent culture media, and obviously then therefore long-term to poor results from when it comes to does the patient get pregnant. So maternal cell contamination is something that really needs to be understood, and certainly the lab culturing conditions, what type of media, how were the eggs cleaned, what type of insemination, how big were the droplets, was the embryo media changed at some point, was the embryo washed during that change? All of those things can lead to discordance, and they're all really important things to take into consideration. So just thinking back to culturing protocol, just to summarize what we've just talked about, we've seen variable non-invasive DNA sample whole genome amplification success rates have been reported, and certainly we're seeing in our own development work. Variable WGA yields have been reported and seen in our work. There's certainly a need to try and avoid maternal DNA contamination through diligently removing cumulus cells after the egg is retrieved, but before it's inseminated and put into culture. It's our opinion that changing culture media somewhere around day 3 or day 4 is really, really important to holding down contamination rates and giving the best results possible. And clearly there are different rates of concordance with embryo biopsy between the published studies. PerkinElmer is currently working on an application note looking at what we think are best practices in the embryology lab around non-invasive PGT-A. That should be published fairly soon. That will be shared with everyone here the minute that it's published or available to you. And it's not the end all be all. It's just what we know today. We would imagine that we're going to learn more tomorrow and the next day and the next day. I think this is a really important slide. This looks at the concordance between embryo stage and cell number. And so this is spent culture media from more advanced culture time points improves WGA yield and concordance with biopsy. So you can see that in looking at things like failed WGA, weak WGA, WGA or results of tests that passed the quality control measures, and then looking at concordance and non-concordance. The later on that you sample the spent culture media, and here it's between day 5 and day 7, then the higher QC pass rates you have, the higher concordance rates, and therefore lower non-concordance rates. You have less weak WGA, you have less failed WGA. So clearly waiting until the embryo has had some time in that media till day 5, day 6, day 7, I think is going to be critical long term to when do you sample spent culture media for non-invasive PGT-A. Here we looked at hatching or biopsy. And so this is clearly showing the DNA yield following WGA of spent embryo culture media. These are all day 6. The yields of DNA were higher if their embryos had been laser drilled during culture. However, again, the yield is variable between embryos. And so even within the drilled zona, while they are generally brighter, meaning there's more DNA there, you can see there's still some variability. So I think for me, this question, the jury is still out on whether biopsy or drilling the embryo before you sample is going to be a benefit, a hindrance, or it's something just we need to understand more. I personally have a student who's doing some work around this question, and I think again another thing that we'll know a lot more down the road. But this is a question that still remains to be seen whether drilling the embryo is a benefit to spent culture media analysis. So how can we then optimize WGA? I've spent a lot of time talking about the things that can go wrong and the things that are in spent culture media that that are not good for PCR. So how can we optimize that? Well, obviously the volume of the droplet, and of course the volume of that droplet that you stick into that you place into your test, that can change or can be optimized. For invasive biopsy, we have a lysis step before WGA because obviously we're putting in intact cells. Actually, it's almost an intact tissue. And so there has to be a lysis step. With spent culture media, we shouldn't need that lysis step. So are there steps? Are there things that we can add to our test that help stabilize DNA or help capture more DNA or just make the test better? Obviously, whole genome amplification technology. Do you use the current form? Do you reformulate? What kind of PCR program do you use? Do you change the PCR program to try and make more DNA or make longer DNA fragments, etc.? You've got library prep and sequencing parameters. Do you sequence more? Do you sequence less? There's lots of things you can change there. And then lastly, bioinformatics pipeline. And I think for me, this slide is really important because again, as PerkinElmer, we have a kit that is designed specifically for non-invasive PGT-A. That kit was developed again in collaboration with 15 labs around the globe. So our kit was specifically formulated changing a good number of these parameters that I'm showing you here. A good number of these things have been changed in our non-invasive kit to try and generate better concordance and therefore better outcomes. If, again, you're using an off-the-shelf kit that's for invasive PGT-A and trying to make that work for non-invasive, or if you're trying to develop your own workflow, that's difficult because you don't have access to the chemicals and reagents that are in the kit. We obviously have access to every single reagent that goes in that kit because we make all those reagents ourselves. Therefore, we can swap in, we can swap out, we can try this formulation. We can do a lot to come to to a conclusion where we have a kit that is, again, created specifically for non-invasive PGT-A, which should hopefully long-term show better results than trying to change an invasive biopsy kit into a non-invasive kit. So where are we today? That's, I think, probably just to finish up here in the last few minutes of the talk, where exactly are we today in non-invasive PGT-A? So again, here's that selection of published studies. This is not exhaustive. This is not every single study. I would point you to, there's a very nice review article by Lever and Wells that came out earlier this year that looks at a lot of papers looking at non-invasive PGT-A. It's a really good read. I highly recommend that you read it. But here what you'll see is varying rates of WGA positive results, varying concordance rates between biopsy and the spent culture media on the sex chromosomes and the autosomal chromosomes, false positive, false negative, and again, there's your references. And you can see they're variable. They're highly variable. This is some work we did very early with the now late Michelle Lane at Repromed in Adelaide. This was at ASRM 2 years ago now. And this is, it's a small study, but highly concordant results between spent culture media and the biopsy, which is really good to see. This is I'm going to just summarize our collaborative study. This is just 8 labs. This is not all the laboratories. These are the 8 labs that had the most results. What we're showing here is the volume of culture media, and I think that should be recognized. Then we look here at average DNA yield, the number that we would consider to be a weak whole genome amplification, the number that were failed, and then autosomal concordance and sex chromosome concordance. And again, what you'll see is variability across the laboratories. Now, some of that variability might be to do with the amount of the volume of the droplets, obviously maternal cell contamination, how well you clean the embryos, do you wear gloves and gowns and hats and masks when you're working with the embryos, all things I think you should be doing. But here again, you'll see that the number of failed the number of failed results is 10%, right? So with invasive biopsy, that number should be more like 1 to 2%. And so I think that's something we all have to understand. I'm as excited as everybody else is about non-invasive PGT-A, but we have to be able to counsel our patients that far more results are going to come back with no result with non-invasive PGT-A, at least for the moment. So in our hands, droplet size from 10 to 60 microliters did not vary significantly. There is some variability there, but they didn't vary significantly, which is good to know. It means, again, our goal with having a kit as a manufacturer is that we don't tell you how to do your embryology. We simply tell you how to collect the samples. We give you maybe some parameters to work within, but we'd like for everybody to be able to do their embryology the way they want to do their embryology. And so this was a really good finding for us. We were quite happy to see this. We also can see that what we would consider a weak WGA in working with what we're used to, which is typical biopsy, even a weak WGA can still produce excellent results. This was 6.4 nanograms per microliter of DNA, which is quite weak, but gave a perfectly lovely normal concordant result. We also tried to break down the results that we have between continuous culture medias and two-step culture medias. And there's I'm sure everybody knows there's a change in the field to going towards these medias where you can put it on at day 0 and you can leave that embryo in that media all the way through to blastocyst. But what we did see is we saw a higher failure rate with continuous culture than in two-step culture. Right, so here the number of weak is about the same. The number of failed is different. 4% in two-step media, 10% in continuous culture. We're continuing to collect data on this to try to understand why that is, but just a thought to go forward with continuous culture, you have to be aware that we do see higher failure rates with continuous culture. We also saw higher concordance rates in two-step culture media with when looking at autosomes. So we averaged about 81% concordance in the two-step labs, the 4 labs there. We were around 45% with the one-step continuous culture. So far in our experience, it seems that two-step culture media gives you higher concordance rates for those, and also higher concordance rates for the sex chromosomes. And again, you can see the data here, 85 versus 68. So just to show, we like to show results. This is spent culture media, sample minus 14 plus 21. This is the trophectoderm biopsy of that embryo. You can see very, very easy to discern the result here. And the spent culture media did really well. Of course, that doesn't always happen. So here's a case with a spent culture media, came back with a clearly 46XX. This group did biopsy with PG-Seq, and you can clearly see the results of this for 46XY. This is clearly maternal cell contamination, and this is the biggest problem in non-invasive PGT-A that faces us right now is controlling and minimizing and understanding maternal cell contamination. I would be remiss in this talk if I didn't bring up just one paper. I'm going to really dive deep. This is a fantastic paper by the group at iGenomics. Carmen Rubio was the first author. This is a multi-center prospective study of concordance between spent culture media and TE biopsy. Carmen Rubio looked at 1,301 paired spent culture media TE biopsy samples from 406 IVF cycles, which is 371 patients. Overall, the concordance for all chromosomes, that's autosomes and sex chromosomes, was 67.7%. You can see the breakdown here. This was an amazing study because all the other studies I showed you, you can see were 10, 20, 30, 40 embryos. This is 1,300 embryos. There's no study larger. There's no study that to date that we will get more information from than this study by Carmen. Again, what I want to point out here, it wasn't just in our hands as we were developing our test. In this prospective randomized trial, you'll see the no result from media was 12.2%. The no result from TE biopsy was 2.6%. And I think that's important. Anybody that's ever seen me give a lecture or ask me the question, I think it's really important that we understand how the test that we're offering to a patient, how that test works in our hands, what is the positive predictive and negative predictive value, what is the false positive, false negative rates. All those things, the number doesn't have to be a certain number, right? As long as you know what that number is and you can explain that to a patient so that you can be sure that the patient understands what it is that they're getting when they get this result. And I think this is a really important paper to start to begin to think about how do you counsel patients around non-invasive PGT-A, how do you produce results and talk about that to a patient. And again, I think one of the things here that has to be explained to patients is that right now, at least somewhere between 10 and 15% of the time, we're going to take spent culture media and we're not going to get a result back, which is going to be frustrating to patients. We need to be able to manage that and understand how different spent culture media is to doing trophectoderm invasive biopsy procedures. So again, just to sum up, where are we today? DNA yield was not affected by embryo culture media volumes, at least not in our hands. Sex chromosome concordance varied between individual clinics, suggesting that embryological processes are important in non-invasive PGT-A. We are continuing to do further analysis of our own data, and we're also continuing the collaboration with a few of the collaborators to look specifically at quality control parameters that would allow acceptance of the non-invasive PGT-A results. So in other words, what QC parameters have to be met to get to the end point where you feel pretty comfortable with that result. Successful non-invasive PGT-A using spent culture media will possibly require specific culturing conditions and or specialized molecular methodologies for accurate and representative amplification and testing of embryonic DNA. It's a lot of words to specifically say that we created a specific kit for non-invasive PGT-A that's very different than our invasive PGT-A kits, specifically because of all the things we've talked about during this talk that can get in the way or that can impact the whole genome amplification and therefore the results. And so one of the things that we've figured out and that we recommend to everybody that uses our kit is that renewing the culture media during IVF certainly improves overall concordance rates between embryo biopsy and spent culture media for non-invasive PGT-A. Even if you're using a single-step media, we highly recommend that embryo somewhere around day 3 or day 4 is removed from the original droplet, washed in new media, and then placed into a brand new droplet of media that it will stay in until it gets sampled for non-invasive PGT-A. And with that, I'm going to finish up. Here are my contact details. You can certainly send me an email to my email address. That's my mobile number if you want to text me or call me or find me on WhatsApp, etc. And I'm going to finish here, and I'll be happy to take some questions. Thank you very much.
Hello, Gary.
Yes. Hi, how are you?
Okay. There is one question from the floor. It would be a groundbreaking if we could do the using the culture instead of the biopsy from the blastomere. And now how far that we can get it implemented now, or it already implemented, this kind of technology?
Yeah, I think that's a great question. Certainly the iGenomics group that last paper that I showed, they are now offering this clinically to patients, not using my kit. They're using their own workflow. There was another group in Australia that you might be aware of that was offering this clinically. Again, not with our kit, using their own workflow. They've had some clinical issues there, and they've stopped offering this test. So, my answer to the question is, I don't know that we clinically understand everything we're going to understand about non-invasive PGT-A at this moment. There are a number of laboratories that are doing validations, and I think that's critically important to do really, really good validation studies to understand the test so that we can talk to patients about it. There's a lot of people in the field that are very excited about non-invasive technology with mosaic embryos, and do you transfer them and those sorts of things. I have colleagues in the field that are saying they'd rather have non-invasive and maybe less results, but at least more clear-cut results. So I think this is going to be pretty common in the next probably 18 months. But today it's not very common that it's not that everybody's using non-invasive PGT-A, but there are certainly a large number of studies and validation studies ongoing. And I think over the next probably 6 to 8 months, you're going to see a lot more clinics coming out with a non-invasive test that they're offering clinically to patients.
And Gary, what is the exactly mechanism? Meaning that the embryo release the chromosome or fragmented of DNA out to the media. That's why we can use the media to do the amplification.
Yeah, so there's some question about where this DNA comes from. We know that embryos, we know that cells inside the embryos certainly go through apoptosis. There's some question in the field whether that apoptosis is directed. In other words, the embryo is deciding to kill this cell over others, or whether it's just random and it just happens. There's some other theories on where this DNA comes from. We now know that there is DNA in the blastocele fluid of a blastocyst as well. So I don't know that we have a full understanding of where this DNA comes from, but it's very clear that in almost every single culture media sample that has been sampled, there is DNA from the embryo in there. Again, I talked a lot in the early part of that talk about contamination. There's DNA from other places as well. There's DNA in the culture media. There's DNA that can get in there from maternal cells, from paternal cells, so sperm and cumulus cells. And then obviously there's lots of places that it can get contaminated along the way. So I think in a short answer is we don't fully understand where this DNA comes from or how it gets there, but it seems to happen in every embryo. It seems to be ubiquitous, or for every embryo there does seem to be DNA in the spent culture media, and therefore collecting it and understanding it should allow us to offer routine non-invasive PGT-A in the very near future, and be able to talk to patients openly and honestly and counsel patients about the pros and cons of this test versus invasive biopsy or not biopsying or whatever it is that patient has questions about.
Thank you so much, Dr. Gary Harton. And time is up, and we have to move on. Bye-bye.
Thank you. Bye-bye. Thanks for having me.